which gain molecules at their front and lose them at their back, so they tend to push forwards on this cell. So they're pushing forwards, and in order to move, they have to grab hold of the substrate and pull themselves forward. If you're going to move forward, you need a net force. Unless you're actually swimming, you need a net force, so you grab the substrate and move forwards. And the way the cells do this is through focal adhesions, which are basically places where they grab hold of the surface and pull. And some of the new focal adhesions come in these lamellipodia around here at the front of the cell. If that was the only thing that was happening, these cells would sort of grab on and move and pull themselves forward and get longer and longer and longer. So they also need a mechanism by which they contract themselves back to circular. Surface tension is one of those mechanisms probably, but also there's a sort of active surface tension, which is that molecular motors move on these filaments around the cytoskeleton of the cell, around the surface of the cell. You have an actin network with molecular motors, and those molecular motors tend to pull on the cell to make it circular again. So, to me, this is my best understanding of a simple model of how cells move. There is a polar force. By polar force, I mean an unbalanced force. This fluctuates, and as a result of that, you end up with a persistent random walk. Very much these active Brownian particles we've been thinking about. But these are squishy cells, so if you just had that force, it would get deformed. So you also have forces which preserve the shape, these balanced forces, these nematic forces that I've been talking about. Plus, probably, some people think surface tension is important there. So, let's just think about reality for a bit. Where do these cells move? These cells move through the extracellular matrix. The extracellular matrix is a messy array of polymers, mostly collagen, and then pictures of it look like this. So typically, if you've got an epithelial cell layer, it'll be sitting on some sort of substrate like that. If you have something like an escaping cancer cell that's trying to get from one place to the other, it's trying to tunnel through this slightly elastic, very complicated system. So the other thing I should show you is now a real 3D cell. These are real movies of a real cell. We're in 3D as opposed to 2D, which we all want to do, and actually look what actually happens. Okay, so this is white blood cells. They're moving through the extracellular matrix. They're trying to show you that this is the old videos, and the ones on the right are the new proper 3D videos, not very new now. Okay, and here it is trying to move through the extracellular matrix. So this is real experimental results. So when you're looking at your nice round or ellipsoidal cell with a few forces acting on it, occasionally one should come back to a movie like that and realize what reality is, that reality is more complicated, and biologists are quite right to complain about physicists oversimplifying everything. But on the other hand, if you don't oversimplify things, if you don't look for generic properties, I mean, you just get a mass of information which you can't understand. Okay, so that's how single cell moves. What I want to mostly talk about is the collective motion of cells. And the more I work on this, the more I don't understand. So let me start by telling you about the phenomenology. What are at least some of the experimental observations that we're trying to understand. I'm going to concentrate on epithelial cells. As I said yesterday, epithelial cells line the cavities of the body. Skin cells are epithelial cells. They tend to have a surface which is next to water or space, and underneath they'll have a layer of this extracellular matrix. This is the picture from Wikipedia showing the sort of structures you can get, and you can see that they like to form layers. They're meant to be a protection often, and so they like to form tightly packed layers like that. So thinking of them in 2D is often not a bad approximation. And this is a picture looking down on one of these epithelial cell layers in reality. So how do these cells move? And it's sort of important because, I think it's one of the really major cell groups in the body, and it's the one where 90% of cancers start. So it would be really nice to understand better what it is about their properties which leads to these cancers, what it is about the properties that we can, if we understand it better, we might be able to do something about it. So 3 things. First of all, there's jamming. Sometimes what happens is that the concentration of the cells increases so much that they can't move relative to each other. And that's called a jammed state, so they just wriggle about their local center of mass. They can take off and flock. This is a picture I showed yesterday. These guys are flocking, and this one I didn't show. This is the same thing. This is Drosophila, a fruit fly egg, and the cells on the outside of the fruit fly take off and go round and round like this. This is a picture of them going round and round, and here it's a sort of 3D version, and those colored dots showed you how they were moving. So that's very obvious flocking, just like we saw with the birds. Nobody understands why these decide to start flocking. One reason is that this is active matter. Essentially, this is confined active matter, and we know from the puppies that confined active matter will go round in a circle, so maybe it's just because it's active, so it happens. Other people think that they move around to extend the egg chamber. The egg chamber starts circular, sort of with a circular cross-section, like a sphere, and then this motion maybe helps it extend into an ellipse. The other thing, as I said yesterday, is active turbulence. This is now showing you a little bit more in terms of why it's reasonable to interpret this in terms of active turbulence. First of all, I've plotted a typical velocity field in these cells. One of these is from solving the continuum equations of motion that I've been on about a lot. One of them is a velocity field from the cells. You can see vortices in both of them. You can see these jets of flow. And if you do a much more careful comparison, things like the velocity-velocity correlation function are essentially identical between the model and the cells. People have found topological defects all over the place in these cells, and they're active topological defects. You can see them being created and destroyed. So again, that's a characteristic of active turbulence. And a very beautiful experiment is what happens when you take these cells and confine them. I think I have shown this, that they start going round and round if you confine them in a box like that, just like we saw yesterday. But a particularly nice experiment, but they don't have a nice movie in their paper, unfortunately, is from the Paris group. And what they did is put these cells in a channel, so they confine them in a channel. And what you end up with is shear flow. So what I'm actually showing here is the flow field. And we know how to solve the nematic equations in a channel. We know how to get that shear flow profile out exactly. So they matched this with an analytic solution. And what you end up with is almost a perfect match. The almost is that the cells are very slightly chiral, which you have to worry about. But, I mean, this was a really beautiful experiment which said these things are behaving like active nematics. In their original version of the paper, they assumed that these were contractile active nematics because that's what you do if you know about cells. Cells are contractile, everyone knows that. They then had to publish a retraction to say that if they got their signs right, they have to use these cells being extensile to match the data. So again, these cells in a rather strange way are acting like extensile cells. I was talking yesterday about this idea that cells can be either contractile and extensile because they can deform and the shape and stress axis can be at an angle to each other. We want to go back to this experiment now and just think about whether we can say anything sensible about this stress shape mismatch in confinement. We just haven't done that yet. We just haven't had time. But it'd be interesting to see how it works in this confined system. Sorry, slight diversion to something I'm interested in understanding at the moment. This is more on active turbulence in these cell layers. Showed you this yesterday. I think we've caught on now that we can take the shape of the cells. These these lines are the long axis, the directive field, which tells me about the shape of the cells. And here's a topological defect moving happily towards its head in a confusing way. But I quite like this graph, which is from that old paper now. This plots the number of topological defects as a function of time. My experimentalist colleagues, in particular Thuan Beng Sor, and this was in Benoit Labadou, led the experiments here. What they did is at the time t equals 0, they added blebistatin to the cell layer. So blebistatin is a sleeping pill for cells. It stops them moving. So they change from an active nematic to a passive nematic. So what happens is that the defects annihilate out, just like they would in a passive system, and the number of defects decrease. Here they sort of put on the cellular alarm clock. They wash out this blebistatin stuff so that the cells wake up. They're not affected by it, and then they wake up. And what happens is that the number of defects increases because now you're getting these active defects created. So I think that's pretty strong evidence that what's going on here is that you've got an active system, an active nematic of some sort. This is one of my favorite experiments. It's from the Milan group, some time ago now. And what they did is they looked at the layer of these epithelial cells, slightly different one, okay? And what they saw is active turbulence. So what's being plotted here is the velocity field. And the colors are the magnitude of the velocity, and you can see it's pretty turbulent. So this looked like the sort of active turbulence you see in these cells. Then they added this chemical, RAB5A. Nobody's quite sure what RAB5A does, but they think that it mucks up the cell-cell junctions. So in some way, the cell-cell junctions are being destroyed. When you add the RAB5A, these cells suddenly take off and start flocking. Nobody quite understands why, but it suggests that cell-cell junctions are important in this system. So here you see the velocity field when they start flocking. And this is the velocity as a function of time. The blue line is the control, the mean velocity in this system. I guess the mean magnitude of the velocity as a function of time when you don't muck around with it. This red curve is when you add the RAB5A. The velocity increases enormously, and then as the RAB5A, whatever it does, wears off, goes down to the old version, to the old velocity. For biological data, even if they don't have error bars on it, for biological data, this is amazing. The fact you have data where the error bars don't overlap and you have a factor of 5 or so here is just amazing. All right? So let's look at a movie of that. The one on the left is the control. The one on the right has this Rab5A in. And what you're seeing is the velocity field. I'll play it twice because it goes on. And you can see it makes an enormous difference adding this chemical. And I like this because it's clear that there's a difference in the two cases. Great. Yeah?
The effect is washed away, right? You're showing the entire window, not only the central part. Since they are basically, you're going back to have some vorticity, or
yeah, I mean.
So you are around the 20 hours mark, right?
The movie's probably here.
Okay.
All right, I don't think it slows down. You're not seeing it slowing down. So this one here is the control and nothing happens.
No.
This one here gets going, and I'm not sure we see it slow down.
You see at the end there. Okay, well.
I think that might be more the zoom than the movie. But yes, I mean, if you look for longer, it would go back to looking like that. Yeah, other questions? Right, what comes next? So, cells are behaving in a different way when you put them in a monolayer, when they're connected to each other. Now, there's something in the biological literature which is called contact inhibition of locomotion, which was noted a very long time ago by Abercrombie. Yeah? And what he said is that if two cells come into contact, then they tend to turn around and move away from each other. And that's been reformulated in terms of sort of modern language, which says that the polarization tends to point away from the overlap between the cells. They tend to not like each other, sort of look at each other and then zoom off again. That has been reinterpreted as saying that if you have cells in a monolayer, so they're next to each other, the polar forces are turned off. So the cells are sitting next to each other, okay? And it would be a bit stupid for them to have polar forces which are all trying to pull them in different directions. And so what happens is that somehow there's a mechanism that when they're in a monolayer, they turn off these polar forces. This is almost a myth, something which has been passed on from generation to generation, so everyone believes it. But if you actually try and look at the literature, the evidence is not clear. And nobody bothers to measure it because biologists don't like measuring simple physical things because they're too easy. Certainly it's true that cells within a colony tend not to form these lamellipodia so much, but they certainly form them a bit. Okay? So probably polar forces are at least turned down in a monolayer, but we're not absolutely sure about that. And again, something which I think needs more investigation and something which we're trying to work on now is that we've been assuming that the polar forces are turned off, and what you're left with, therefore, is the nematic forces. Makes sense because the neighbors will be pulling on a cell in this sort of way. The cell will be trying to pull itself back to circular. So what might be happening in the layer is that these polar forces are turned off, and what you're left with instead is lots of things pulling on each other in a nematic, in a balanced way. But there's also some really nice work, a really nice idea by Cristina Marchetti, who says that what you get is fluctuating polar forces, right? You can't have a direct polar force in this layer. You've got to have fluctuating ones, because else it would flock. And fluctuating polar forces certainly might be happening. And if they are, then if they're fluctuating very quickly, they're going to look nematic, because you're going to average out over the directions, and then if they're fluctuating more slowly, they'll go back and look polar again. Yep? No, yeah. And let me just say, when you say just to be sure, what I'm talking about now is very much at the forefront of research. You know, I might be wrong. We're still thinking about it. So questions are great because it will help me think about it.
Yeah.
So, yeah.
No, it's more a technical question about the definition of polar in the monolayer is just like the production of lamellipodia.
Yes.
Okay.
Yeah. Well, that would be one way of doing it. Yes. It's
pulling
pulling on the surface.
Okay, so traction
so
force microscopy.
Traction force is another way of putting it. It's something where you need something to push against because you need a balance. So this is pushing on the surface. This can happen even if you don't have a surface because they're just pulling on each other.
Okay. Thank you.
I've just been arguing with one of my graduate students about where this pneumatic force comes from. It could certainly come from cells pulling on each other. What she was saying is that maybe the cell pulls on a substrate and then the substrate pulls on the neighboring cell. And that could be true as well. So we realized we were switching these around horribly. So we're still thinking about this. I do not think there's anything super clear in the literature. Yeah? You can comment.
Showing later an experiment I've done with these people later, where at least in this flocking regime, you find all the signature of flocking physics.
Yeah, I'm sure that's true.
So, I mean, of course, the moment that they got this Arabe drag, they really went to show polar forces, I think.
Yeah, I think once it's doing this flocking stuff, once you put in the Rab5A and it moves off, or in this Drosophila it's going round and round, then it's polar forces. I think that's pretty clear. I'm thinking about this active turbulent light regime, where it's going to be either sort of straight pneumatic forces, but it might be quickly fluctuating polar forces. Okay, other comments? All right. So then I was talking about this in Boulder, at the Boulder School this summer, and I was saying, you know, these things do active turbulence. And, oh gosh, I'm going to forget her name. I'll get there. What's the lady at Chicago called? Thank you. Margaret Gardell said, and she's a very, very eminent experimentalist, my MDCK cells don't do anything. They certainly don't do active turbulence. They just sit there. Okay, and then we found this paper, and what this paper did, it's a really sweet paper. It's been put in a totally obscure, I think, journal very recently. I was looking at some other work that this group has done and found this by mistake. And what they did was look at a substrate and what the coating on the substrate was different in two halves of the substrate. So this side they coated with ecadherin, which is something which holds on to cells quite hard, where cells form bonds with the substrate. This side they coated with a traditional extracellular matrix sort of protein. From our point of view, just different sorts of substrate. And then they measured the mean speed across the substrate. On this side, the cells wriggled about a bit, but didn't go anywhere. On this side, they did active turbulence. And the crossover was dead sharp. Okay? The ones on this side just sat there, the ones on that side moved. So it really matters what the substrate is, how these things move. And that really has not been taken into account in the experiments. So we really don't have very careful, very simple experiments, because biologists are not interested in very careful, very simple experiments. I'm sure they're careful. Okay, so the message there is we really don't know how these cells move, but sometimes in the right circumstances, they can do they can do active turbulence. Now, can you wake up? I'm sure you're all awake, but I want to model these cells. All right? What sort of questions do I need to answer? So please, can you talk to your neighbors and make a list of the questions I need to ask if I'm going to try and model these cells? And in particular, let's concentrate on them when they're doing active turbulence. You can move if you don't have a neighbor, okay? So I want, please, a list of 5 or 6 really nice questions. And see if they're the same as my questions. Or of the theorists. They're generic questions which we might address by experiment or by theory. Yeah. They're things I ought to be able to explain to you in the rest of this lecture, which I won't be able to, but it would be nice if we could. Okay. Then tell me your questions. Yep, go on. Could shout, maybe.
How do stresses and shape correlate? So stress and shape. There's
That's
information about contractile and extensile.
Yeah. That's maybe a question because of what I was saying yesterday. Before we worked out this shape-stress misalignment, I don't think anyone was asking that question because everyone was assuming they were in the same direction. So the question used to be, till about 3 months ago, are they extensile or contractile? But I think other ones? Maybe even simpler ones than that. The role of cell adhesion. It's a good one. Whether it dominates over other interactions in a monolayer.
So mine is, do gaps form between them? So do they detach from time to time?
The role of gaps, yeah. Yeah, go on.
If CIL is present in dense systems, because ideally it explains it explains low density like interactions, right? Only one-on-one cells, but in high density I don't understand it.
That's right. The original story was when you have just 2 cells, and it's sort of been almost in folklore been made into what's going on in lots of cells. Yeah.
I would say also how heterogeneous is the stress field on the monolayer, and how like the dispersion of the velocities correlates with local density also.
Okay, so density fluctuations, density and stress fluctuations, yeah?
And correlation between them.
Anybody else? Mine were much more simple than this, actually, so don't be scared of saying simple questions. You guys at the back were discussing nicely. What did you come up with? Yeah, well done.
Maybe if the cell is in a reproduction phase, like is
So the effect of cell division.
yeah.
Yeah, true.
Here we talked about viscosity. Maybe if it's possible to measure it.
Effect of viscosity, yeah. I think you're missing out the really simple ones. All right? I mean, those are all good questions. Those are the ones I had. If I want to model this, what are the right forces to put in? We've talked a bit about that. And even if we put them in, what's the biology that's giving us that? The effect of the substrate, that's, to me, is a very, very simple first question. If you've got edges, is it going to be different at the edge of a cell colony than it is in the middle? The squishy cells I've talked a bit about. And then this thing at the end, extensile or contractile there. But your questions as well are all ones we would like to answer. So we've got to, if we're physicists, let's do some models. What sort of models do we have? But before I do the models, let me just remind you that we are physicists, but physicists sometimes don't appreciate how complex biology is. All right, so I think that is, there's a really good review of models, if anyone's interested, by Ricardo Lert and Xavier Trepat, with that reference there. We've talked a lot about continuum models. I've talked about active pneumatic models. One could also have polar models. In Paris, they call them active gels, but those are the same as active pneumatics. We've heard a lot about particle models and the V-check model, and other people are much more expert on those than I have, and I've told you about those very nicely. There's a thing called the cellular pots model, which I dislike intensely because it's obvious that the physics is wrong. So I'm going to talk about, first of all, phase field models, which I also have got to dislike a bit, but that's just because it's really hard to do the computations. And then I'm going to talk about vertex models, which I've been learning about due to the fact I've got a super postdoc, Jan Rosman, who's an expert in these. So continuum models are great. They're probably the easiest way to look at these things. But if you want to ask questions like the role of cell adhesion or the role of gaps, you've had it, because you can't resolve individual cells. So let's go down a length scale and look at cells when we do resolve the individual cells. And one way to do this is a thing called the phase field model or the multi-phase field model. And this is the sort of simulation that you would see doing a phase field model. Basically, it's a bubble model with activity. So let me explain a little bit how this model works. So each cell is described by a phase field. This phase field takes a value 1 inside cell I, and it takes a value 0 outside cell I. So it's a marker for where that cell is. The phase field evolves in time according to this equation. So this is the normal advection term. It moves with the velocity and at the same time, it relaxes to a minimum of a free energy, and I'll have to tell you what that free energy is. So these cells are moving around with some velocity, and they're also relaxing down to the minimum of a free energy. Not so different from the continuum. Then, at least in the original version of the model, the thing is overdamped. So we say there's a force acting. I just choose what the force is. Okay, I have a force acting on this system. We're going to have forces because it's minimizing its free energy, but I can also put on active forces. I could put on an active polar force. I could put on an active pneumatic force. And then I just say that the force is proportional to the velocity. It's moving on the substrate. All the momentum is immediately dissipated in the substrate. This is what you call overdamped dynamics. It's a completely dry system. So what about the free energy? Well, now I'm going to list the terms in the free energy. First of all, you have a normal Cahn-Hilliard term, which I hope you're used to. This thing is a double well potential, which says that my phase field variable phi I either wants to take the value 0 if it's outside the cell, or 1 if it's inside the cell. So this thing is a very traditional double well potential, and then this thing here is a surface tension. It says that where it's changing from 1 to 0 at the edge of the cell, I have an interface, which is a diffuse interface, which changes continuously from 1 inside the cell to 0 outside. And then basically I choose the term in the free energy to get what I want out of it. I'm just guessing to make sense. So first of all, I say, okay, I want all these cells to have more or less the same area. And so I have a soft constraint on the area. This one here, it just says the area here needs to be about 1. So I choose my radius r for the cells, and then their radius is more or less r. Then I don't want them to sit on top of each other, right? I want them to sit next to each other, and so I penalize regions where they sit on top of each other, so phi doesn't like to be 1 for 2 neighboring cells. But on the other hand, I don't want them to fly off, so I've got loads of space between them, so I also need an attractive interaction, the adhesion interaction, and I mock that up by saying that the interfaces like to lie on top of each other. So there's nothing amazing about that. It's just let's get it looking like a cell monolayer. Let's choose the free energy so that in equilibrium it comes to look like a cell monolayer. And indeed, in equilibrium, it looks like a load of identical hexagons sitting next to each other. Sometimes you can get jammed in a metastable state and it doesn't get there, but that is the minimum of this passive free energy. So you might as well use this to describe a load of bubbles as a load of cells at the moment. So what makes cells special? Well, they're active. So we need active forces in there. We've been talking about polar forces, and we've been talking about pneumatic forces. So let's put a polar force in now. This is the sort of force you'd put in a model if you wanted to flock. So what we do is we choose a polarization force, and there's several ways of doing it, but a very simple way is to just say there's a vector associated with each cell, a direction associated with each cell, and then this is the phase field, which tells me where the cell is, and that alpha is a strength of the polarization force. So by writing this, I'm just really not doing anything except saying I've got a force on this thing. It has strength alpha in a direction pi, which I call the polarization. And you could have it acting on the center of mass of the cell. You could have it acting on the boundaries of the cell. Here I've got it spread out all over the cell. And you could choose that polarization to work in many different ways. You could just have it going randomly So you've got a sort of active Brownian particle-like cell. You could make it align with the velocity of the cell. So the direction this thing is pulled is aligned with its velocity, and then you'd have a flocking model, you'd expect. Or you could assign it with the shape of the cell. These shells are going to be extended in some direction, and you can line this polarization axis with the shape of the cell, and you can change its magnitude. You could say it was 0 if the cell is circular, but it increases as the cell gets more elongated. And I don't think we actually know what happens in real cells, so what we thought we'd do is we'd just try all of them and see what difference it makes. So let's see what sort of results we get when we run this model with these sorts of polarizations. So we're really looking at a dry flocking-like model. So we're not doing something very different from active Brownian particles. The big difference is these things are connected to each other. They're joined to each other by something which is acting, we hope, like a cell-cell junction. Oh, just with this alignment one, I have to have a time scale, you know, how quickly it aligns, and this is the time scale that we use. Any questions about the model?
I have a question, but you mentioned, okay, you mentioned that you don't like phase field models very much because they are hard or something like that. What are the technical difficulties that arise when you're...
The numerics.
The numerics.
It's really hard to do a lot of cells.
Uh-huh.
So we can do about 200 in 2 dimensions, and we can't do it in 3 dimensions, and even 200 takes a long time. So that's why I don't like them, because the poor graduate students go wobbly because of this.
Yeah, thanks.
Okay. And I thought they were much nicer than vertex models, because vertex models are, you know, polygons, and these things have nice round boundaries. But we've been trying to do the same calculations on these phase field models and on the vertex models, and we get the same answer. So on vertex models, you can do 10,000 cells really quickly. So I've been a total convert to vertex models. But it's good to always check lots of different models, because if it's generic, you've got more chance. Yeah.
Julia, sorry.
Yeah.
Go.
Okay. Julia, here.
Hi, sorry. Yeah.
That's okay. Yes, I have one comment. Tomorrow, maybe I'm going to comment something on these continuous models, on how they are modeled numerically, and why it's so difficult, why it's so hard.
Hang on. The continuum models, as in the hydrodynamic models, are not difficult.
No.
But these guys, yeah.
Yes, because of the cangiglier, that is a double ablation. That makes things quite hard.
Yeah.
But I have a question. How do you obtain the long axis of the cells here? Because you said that you're going to align the polarity to the long axis.
You have to essentially have a shape tensor for each cell.
Okay, so
so you're
using
assuming
the same phi field.
Yeah, so from that phi field you get a shape tensor for the cell, and then you get it out from there.
Okay, cool.
Yeah, I always get confused with the phase field model. It's how you define each cell. You basically have one phi for each cell.
Yeah.
But then you also have like the pixels inside of each cell.
That's right. That's really why it takes so long, that you have a phi which defines the inside of the cell, but then you have to keep track of the interface. And okay, you could just keep track of the interface, but the interface is moving, so where you keep track of is also moving in time. So we've got the whole thing on a very well-resolved grid underneath, and each cell, you have to sort of do a grid around each cell, and it's horrible programming.
And then the area term is necessary, so you kind of conserve the number of pixels then?
The area term is necessary, no. Just because physically these cells are going to have, they're not suddenly going to get huge or tiny. But the pixel story is a different problem.
Okay.
Julia, I see that your model is not conserved, so you are assuming that the areas of the cells can change. It's not a conserved quantity.
Yeah.
That's right?
Yeah. And that's physically very much what happens. These things can shrink or grow, but it's partly because they're actually three-dimensional. So what they're doing is being big and not very tall, and then when you see them shrink, it's usually because they're getting bigger in this direction. The actual volume is conserved to a good approximation.
Okay, thank you.
Okay, anything else? Okay, let's do it. So this is what happens with polar forcing. What happens is that for almost all those different possibilities, you end up with a jam state. So what I'm plotting here is the center of mass position of those cells, all right, as a function of time. So this cell is around this point here, and it just wriggles around a bit, so its center of mass moves a bit, but the cells never change relative to each other. So that's with small forcing. Or you can get a state which we've called a liquid, where the centers of the cells move around, but move around randomly. You can get flocking. I think that's a movie showing the flocking. They can sort themselves out and flock, but that only comes in if the polarization is chosen to align with the velocity. If the polarization is random or aligns with the shape, you don't get that. Yeah?
Yes. When I first saw that video, I think some years ago, I don't know, I had some impression, now I have the same impression that the rotational symmetry is broken, and you have a preference for either X or either Y alignment on the order of the things.
I mean, I'm not sure that we did it millions of times to check. We do have an underlying lattice, so it could happen.
It could happen.
It could happen,
okay.
yeah. And I agree, we didn't check that super carefully. That's a good point. I mean, it might just be luck, but we should have perhaps checked it more, yeah?
Could you explain why having a lattice would break the symmetry?
Just because the lattice is like this. Yeah. All right, so this is a computational problem. The flocking will choose a direction, and if I did hundreds of simulations, that direction should be random in space. So Rodrigo is quite right that it's a bit strange that it's top to bottom. And we may have a problem due to the fact that our lattice has a symmetry axis in that direction. We don't see that in, I think, almost any other simulation we've done. So it's not a big problem, but it's something we should probably check better. Yeah. Okay. So then we said, great, you know, this is a cell layer, this is flocking, that's jamming, this is active turbulence. So we looked at this very carefully, and the wretched thing had no topological defects. It was a perfectly good liquid. It was not active turbulence. We are not reproducing what's going on in these cell layers in terms of active turbulence. And maybe that's all right, because we've got a polar force. So let's try and put in a nematic force. So we're turning this off now. We're turning this polar force off, and instead we're putting in a nematic force. All right? Look it up in the paper if you want to know exactly how, but the hand-waving thing is, well, I've written the equation here, is that basically we put a minus zeta q term in on each cell. So you can put a nematic force on these cells, and you could imagine a nematic force inside a cell for a start. So I'm going to show you 4 different possibilities. So let's start with a nematic force. Here's my cell. Here's an extensile-like nematic force. What you'd expect is that the cells are extended, so they're nematic. And if you look at them, you get, again, a state where things move around. You can see topological defects. you can see active turbulence. And that makes sense because these things are nematic and you see active turbulence, you see extensile active turbulence. I'm not going into this in super detail because it's a bit technical. If I put a contractile force on, what's going to happen is that these things are going to squish back to circles. Okay? I must say, we did this before we knew about this stress-shape misalignment. So what we did is that we assumed that once they'd become circles and were trying to do this, we changed the direction of the force so it was still contractile, right? This was 2 years ago before we caught on to that. So these things go back to circles and nothing moves because they're not extended. We then tried to look at interactions between cells. So what's happening is, again, you get dipolar interactions, but now it's across this boundary, so the cells are pushing each other. And what you find is that if you have contractile forces between cells, the cells elongate. That's because essentially the surrounding cells push on the central cell, and they push it in a way shown by these arrows, and the cell actually elongates. And what happens is that, therefore, you get nematics, and these things form a nematic-like lattice. If you put extensile forces between cells, the cells elongate, but they want to line perpendicular to each other. And if you do rod simulations, that's what you see. You see this thing called a capped line state, and I'm saying this perhaps a little bit more for the experts, right? If you have intercellular interactions, the cells really like to line up like this, so you get lines of rods, and then the ones here want to be perpendicular. The cells like to line up perpendicular to each other. So extensile interactions between cells give you this state with what essentially is quadrupolar symmetry. And we can prove that analytically. All right, so that's a quick pass through the through that phase field model. Wouldn't recommend it, really. Takes a long time. So let's think about the vertex model. And I say I'm just learning about this, so I'm sure there are people in the audience who are much better at it than I am. And it's Jan Rosman, who's a real expert on this, who's taught me everything I know and has indeed provided many of these slides. And Jan looks like he's just come out of prison in this picture. I must get another picture of him, actually. But it's tough being a postdoc at Oxford. Yeah. All right, so the vertex model, again, is trying to resolve cells. And it does it in a much simpler way because it says a cell's a polygon, and you have a load of polygons which are stuck together at junctions. You have a term which is an energy term which says that I want these cells to have a certain area. So this is very like the shape term in the phase field model. These cells have a trying to get to an area A0. And you also have a term here which is a perimeter term, which says that they have a preferred perimeter. And so what you have is a competition between them wanting to have a certain area and have a certain perimeter. And then the dynamics is essentially similar, all right? This is essentially my free energy functional that I had before, except it tends to be called an energy here. You have overdamped dynamics relaxing towards the minimum of the energy, and then you add active forces. And the most obvious way to add an active force is to add it at these vertices. So you tend to get a random active force acting on the vertices. And it works surprisingly well in terms of looking at a crossover between a system where these things are jammed. So again, this is the center of mass of the cells. They're jammed here, and you can see they sort of wriggle around. That's what's called the solid phase. And here, then you can get them moving relative to each other, and you end up in a phase which is called a liquid phase, or fluid phase here. All right, and so this is a very similar crossover that we saw in the phase field model. And that's really why this model has become famous, that it can get this jamming transition in cells, and it gets it at a value of little p0, which is related to the perimeter over the square root of the area that you want it to get to. And it turned out that this thing here, which was, I think, 3.81 in the original paper, fitted very nicely some results on cells. So everyone thought, great, you know, this is universal, everything's okay. But now they realize that this is very model dependent and that the experiments are even more model dependent. So you do get this crossover between a jammed state and a motile state, but quantitatively, this is not right. You can also get flocking if you put the active force on in a way which tends to pull these things in the same direction. Yeah, we've got an expert here. Yeah.
Transition. So if this is really fluid, or the neighboring relations stay fixed?
The fluid thing, you have T1 transitions here because you've got the cells moving relative to each other, and so you have to have T1s to make them move.
So there is a little bit of fluidization.
Yes.
Okay.
Oh, yes. Yeah.
No,
I mean,
since it was almost looking like a rigid translation with there is more.
No. In this phase, right, T1 transitions are what's doing it, so it's more like a liquid.
Yeah.
In this phase, where the active force is a polar force, I assume linked to the velocity, but I can't remember, right, then it's much more the whole lot is moving together.
Okay. So
this is
it looks a little bit more like a polar solid.
Yes.
Oh, okay.
Absolutely.
So this is not.
Yeah, you can get a polar solid,
okay.
You can get a polar liquid, and then you can get a much more random liquid. Yeah. Okay. And this thing's great because you can do 100,000 cells if you want to. It's much, much easier computationally than the phase field model. So I want to, hang on a minute. I want to ask, the direction we're going now, and I think everything I'm going to say from now on is pretty much the last year. So I'm now talking about research rather than a body of knowledge everyone ought to know. So do ask questions, because it's great, because then I'll know what the next problem is. We've got this vertex model, we've got the phase field model, we've got the continuum. I'll concentrate, I think, on the vertex model and the continuum. They're both, at the moment, I mean, it would be nice to get the same answer out of the vertex model and the continuum, right? We're meant to be doing the same physics. It would be really nice to set up a nematic vertex model and show that it gives the same thing as my continuum equations of motion, which we're trying to describe a nematic. So we tried that. We put a Q-tensor into this model, again, just putting on forces which look like the ones you get. And what you end up with is something which is sort of looking like active turbulence. This looks on a cell level, okay? And you can see topological defects, and it looks like sort of weedy active turbulence. These things are always a funny shape, which is a bit of a problem. So then we thought, well, it's a bit hard to see what's going on. Let's look at flows in a channel. We know that these active systems must flow down a channel, right? That works in experiments, which is good. It works in the continuum simulations, it jolly well ought to work in this nematic vertex model if we're trying to describe the same physics. And so we did it. We put it in a channel. Those colored things are just so you can see what's going on. The flows just don't develop in the vertex model. The vertex model is missing some of the physics in the continuum. So what's it's missing? Well, the answer is that if I consider a dry vertex model, all right, forget about this term, this is the basic dry vertex model, an overdamped vertex model, any momentum is immediately dissipated to the substrate. You can't get flows. The correlations are not large enough between the cells for you easily to see flows in this system. Not flows developing from nematic order. You can from forcing it, but not flows developing from nematic order. So we tried to write down a wet vertex model. That means that the momentum is dissipated through viscosity, like in a fluid, rather than immediately to the substrate. The momentum spreads out across the fluid, and so then it's dissipated at the boundaries or just spreads out everywhere, like in a proper fluid. So if you put a wet model, which just means that the dissipation is related to the velocity of neighboring vertices, and then look what happens, right? The one at the bottom is the wet model, should look much more like the continuum model, because the continuum model is a wet model. Up, move. Oh dear. This is my punchline being mucked up by the Zoom. Let's see if I can get it to do it. Right, so this one's a dry one at the top, and it ought to move, not move at all. This one at the bottom is a wet model. And what you should see, if you're lucky, can you see the flows moving there? You can see that purple thing. It's not working because of the Zoom thing. This flows beautifully down the channel. You have to believe me, okay? It flows down the channel, yeah?
How do you treat the boundary? And this is one question. And the second is?
The boundary is just you have fixed cells on the boundary. And then they're pulled by the flows. So they look like that. So even here you get very local flows near the boundary because of these things, because of the symmetry of the surfaces, but no flow in the middle. Here you get flow all the way across the channel.
And is there any pressure difference, or is just active?
No, it's active flow.
It's active flow.
Yeah,
okay.
It's just active flow. So because you've got this channel and it's active, it can form distortions in the director which are driving the flow. Yep.
And do you get a parabolic velocity profile?
It's not quite parabolic, but it's close because it's between that. Sorry, I didn't have the pictures, but they are in the paper. That, yeah. Okay, and the reason is that if you have a wet model, you get correlations. This is the correlation length of the velocity correlation length. This is what happens in the dry model. The cells, neighboring cells don't talk to each other. This is what happens in the wet model. You get a much longer correlation length, and that's why it's working. So you need a wet model to map onto what's happening in the continuum. There's still lots of work to be done to look at that mapping more analytically, and we're now trying to make the phase field wet, because I think that's going to help, because one of the problems with this phase field model is it takes ages to do anything. Okay? If we make it wet, the dynamics is going to be much more quick. It's going to move around much better, and we're going to be able to hopefully do larger simulations with it. But that's very much ongoing work at the moment. Okay, that got a bit technical near the end, but do people have questions? Yeah?
What
determines like the amount of sides each vertex has? Some have 5, some have 6. Does it have any meaning?
That's right. In equilibrium, they all have 6, but because of the dynamics, you can get 5, 7 pairs.
Oh, so it has like defects with fewer neighbors and more neighbors?
Yes.
Oh.
Yes. Yes, that's right.
Yeah, thanks.
Okay. So these models are fun. Let's just look at reality. So now I'm going to move on to talk about what actually happens in vivo. And this is what we want to move towards modeling. This is what people are trying to do, and it's hard. All right? The reason it's hard is things like this. I hope the movie's going to move. This is the fruit fly. This is Drosophila. And what you're seeing is individual cells of a baby Drosophila growing into a grown-up Drosophila. You can see correlated flows of these cells. You can see changes in shape, and you can see patterns forming. That's what we want to explain. It's pretty obvious that this is active matter. It's pretty obvious it's not active turbulence. All right? So how does that work? And that, I think, is probably one of the big research questions. How much of it is physics? Some of it's obviously physics because things are moving around. But something has to start the physics off, so we need to worry about the chemistry, and then what is going to drive the chemistry is the genetics. And so we need to put all those things together if our models are going to be useful to describe real biology. Right, I'm going to finish with, I think I'll miss this bit out. And I'm going to finish with the thing I didn't get to in the colloquium yesterday. And this is very recent work, so this is almost a bit colloquium rather than lecture. Your input would be great, because I think it's only the second time that I've talked about it in public. It's not published. And both in terms of trying to explain it properly, so it's nice to have questions to see what I'm not saying, and also in terms of what we still need to look at, it's very helpful to have the feedback. This is work done with a student, Saraswat Bhattacharya, who's just graduated. Well, he will, when he's done his corrections, he will have graduated, and he's going to work in Dresden from Christmas. And the data collection and analysis were due to Joseph Kass in Leipzig and his student Pablo Gottheil. And what Joseph did is realize that there are a lot of data in medical records which might be useful in understanding what goes on in breast cancer from a physics sort of viewpoint. So the way breast cancer works, it's a little bit icky, these things, it's not fun, is that it starts as what's called a tumor in situ, which is basically a tumor of malignant cells which have a boundary due to a sort of membrane about it. And at this stage, it's fine because the malignant cells are stuck inside this basement membrane and they don't get out. And as long as it stays like that, everything is fine. But then at later times, you can end up with the breast cancer being invasive, and it forms much smaller, irregularly shaped clusters of malignant cells. So this would be a cluster of malignant cells, the dark purple ones, with this extracellular matrix around it. And if you zoom in, what you see is that the cluster of cells looks like this. And the green lines show how the cells are moving. And then round it is the extracellular matrix. And the fibers, the polymers in the extracellular matrix, are well known to have local nematic order. So these are the directions of the polymers in the extracellular matrix. All right, so what we wanted to do is that we have loads of data which basically give you cross-sections. Sorry, I didn't have it on there. Cross-sections looking like this. And loads means loads. There are this many cross-sections taken from patients. So it's a two-dimensional cross-section across a diseased breast cancer sample. This many cross-sections, about 10 to the 5th clusters. So that means that when you do data analysis on it, you don't get error bars with that much data. So we can measure things like the sizes and shapes of these clusters and get amazingly good curves. But the trouble is that these images are static because these things evolve on a time scale of years, and obviously you can't do the dynamical simulations. So can we model something and match it to these static images? And both Joseph and I sort of looked at this and thought, that's looking like an active nematic. And these are cells. So we expect the cells to be an active nematic, okay? So let's model it as an active nematic in a background, which is the extracellular matrix. And you expect the extracellular matrix to be a passive nematic. And it's nematic because the fibers of the extracellular matrix tend to line up as a nematic. And you expect it to be viscoelastic. So to do the dynamics, we use the continuum model. Back to the continuum model now. The cancer cells are an extensile active nematic. The extracellular matrix is a viscoelastic passive nematic. We have a simulation which mixes the active and the nematic, so it's like a two-phase thing. And what it does is it phase separates into two different phases, into cell bits and background bits. And because we're doing these equations of motion, we can see the dynamics. So this is the sort of picture. We'd set it off like this. And this is the sort of dynamics we end up with. This is the steady state dynamics of our model. Just to check, we set it off with a different initial condition, which was just a blob of the active nematic inside the passive nematic, and it breaks up, and you end up with exactly the same dynamical steady state. So if we model it like that, looks like we get pretty much the right patterns, but, okay, do you want to ask a question? Yeah, go ahead.
A very quick one. Don't you have to worry about cell division? Or is that an ingredient that you have to incorporate, cell division?
Cell division, no. And that's, that's an important point, which I'm going to come to in a minute. So just remind me to make sure I say that. Because you're right. You're thinking cancer, cancer growth, cell division is really important. These tumors are growing. That's what everyone thinks, right? There is no cell division in this, and it works beautifully. And then we tried to see how much effect cell division would have just by sort of order of magnitude estimate. All right? Will cell division make these clusters move because they're growing and therefore they're moving? And it looks, order of magnitude means that the data is not there to get really good estimates, but it really looks as if motion due to activity pushing them around is more important than motion due to cell division, which is one of the punchlines, actually, of this, that we don't need cell division to see this setup. We just need these cancer clusters to be what we know as active, but the medics haven't a clue. Right. Other questions? Yeah? There's one at the back there.
So in real time, how long does it take? So it's me first, sorry. In real time, how long does it take for it to go from the initial state to the steady state?
We don't really know because that process we're not modeling. What we know is that in the steady state, these cells are moving microns per, microns per hour, 3 microns per, I think it's hour, sorry, I'm not sure it's hour or day, but very, very slowly, yeah. can look it up. We've got those.
So just a technical question. In the simulations, you choose to a contractile or a extensile behavior, or
we assume the cells are extensile.
Oh, okay.
Yeah.
Thanks.
Okay. So it looks the same. Can we do better than just it looks the same? Well, we've got all this data on the static images. Let's take static images from time steps of the simulations, uncorrelated time steps of the simulations, and compare. Oh, sorry, this is just... sorry, can I just go back 1 minute? Forgot about this bit. See, I haven't practiced this so much. This is just time shots of what's going on. What you see is these clusters form a steady state because they're continually coalescing and breaking up. The black arrows show them breaking up. The activity is stretching them, just like we had in the phase separation thing, and that's causing them to break up. At the same time, they're hitting each other, the red arrows, and they're forming a single cluster. So that's just the dynamics. Now, I hope, and this is just pictures comparing what's happening in the simulations to the experiments, in the simulations to the experiments, to the data, really. And you see the same sort of thing. But, it really is at the moment same sort of thing where we choose these to match because we don't know what the activity is in these samples. Yeah?
3 dimensions. Your simulations are in 2 dimensions, right?
Simulations are in 2D. We did a few simulations in 3D, and cross-sections behave in the same way.
Okay.
But certainly the data is 2D cross-sections of 3D, and we did worry quite a lot about that.
Yeah.
It makes small differences, but nothing major.
Got it.
Yeah.
Thank you.
Right, so we actually do get the data now. Yeah, Francesca? Yeah.
Do you know if during cancer you can start like from a carcinoma, like the big cluster which is healthy at some point, and then if it breaks and causes the cell the cancer evolution, or so I'm wondering if in your simulations you can start from a carcinoma and then study how it breaks, or like with protrusions, or
Yep.
yeah, if you have an idea.
That's something I want to do. Here, that was just an initial condition. It's not physics, it's simulations. So we're only really looking at the steady state, all right? Okay. And I don't know enough about the medical stuff yet. I mean, when it's a single huge tumor, you're okay, but it's when it's broken up that you have to really worry. Yeah, that's me.
Thank you.
Mm-hmm.
Sorry, you seem to match pretty well this.
Well, I haven't talked about it yet.
Yeah,
but
okay.
yes.
Yeah, no, but okay. So to talk about that first. Sorry.
Yeah. Okay. Right. So this is the match of this amazing amount of data, which is red, and our simulations, which are blue. The data is a power law. This is the size distribution. Doesn't match here. We understand that. That's because we have a diffuse intermace model, so we can't look at tiny clusters in the simulations. We get a power law. That actually says very little, in fact, because we average over activities, so what we're actually doing is averaging loads of exponentials, and you always get a power law. This is much more, I think, believable. This is the shape index. These are special pictures in case a medic reads it. This is a circular shape. They get much more tortuous here, and the shape index matches very beautifully between the simulations and the data. And this strong correlation between size and shape comes out in the data and in the simulations, and that, I think, is the largest evidence that we might have the right physics here. And so our punchline is that physics, which just looks at these things as active clusters, that the activity is causing this very distinctive pattern of clusters in cancer. I mean, this is what the medics do to, to see what's going to happen to you, basically. This is the medics say this is a very distinctive pattern. We reproduce it with this model. Maybe it's a new way to think about things. Just the last slide is that you can look at defects in the extracellular matrix, and they tend to cluster around the cancer clusters. They're found much more close to the cancer clusters, both in the simulations and in the experiments, again, tending to suggest that you're getting stressors from the active cancer clusters. If you look at this in situ stuff, which is not malignant yet, you don't get this upturn in the number of defects. So I thought I'd end with a picture of where I work. We're very proud of these blackboards. We have them all over the place so we can discuss with each other. That's my research group working hard while I'm here having a nice time. Okay, and thanks for listening and questions.
Okay, thank you very much, Julia, for this wonderful course. So we have questions now.
Okay, so now I can make the question.
Yeah.
Now, I was wondering, how difficult is to reproduce this kind of curves?
The power laws.
The power laws according to the parameter of your model. So if you want, is it possible to deduct, at least in the model, some mechanical activity parameter from matching with the experimental data, or you think quantitative relation are out of questions?
We tried to estimate the parameters from the experimental data that exists. For example, the activity you can get from the velocity of the cells and so on. And these are the best guess parameters. It's not easy because you're trying to match everything, right? Yeah, yeah. And we can match almost anything, and in Lattice-Boltzmann you can never match basically the pressures, right? So it's not bad in terms of matching to reality, but it's not bad, but it's not, it's not an engineering thing.
Okay, do you think it could be useful to biologists? I mean,
in terms of matching?
Yes, or that we can go, I mean, you can go and tell them, look, this kind of behavior is produced by these and that mechanical properties of the cell. If you're able to affect them, you can... Do you think it's a viable path? Since, well, of course, I don't know. I know that no one is still there, but do you think
I mean, I think that's the direction we want to go. But just getting over the message that cells are active, maybe active matter physics matters, is the first step. Yeah.
Thank you.
Yeah, no, I just wanted to comment that it's. One has to be really careful when looking at 2D slices of a 3D object, and I think it is the reason why you see a power law. So if you imagine that all clusters were spheres, but located at different positions in the set stack, then you don't get a uniform distribution of areas, right?
That's right. We did that. We called it the Easter egg thing because we did it as Easter and we were cutting across Easter eggs. It doesn't make a quantitative difference.
In the areas,
a qualitative difference. Sorry. Yeah.
In the distribution of areas, it should.
No, because we're averaging. Probably because we're. I mean, I've got the calculation which we can look at if you like. What we did have to do to get that power law, right, and this is probably an important thing, I think, is that we are averaging over activities, because obviously in the slides, they're all going to have different activities. And it's quite hard not to get a power law if you're averaging over activities. So one mustn't put too much weight on that.
Thank you.
So the vertex model, for instance, is good for confluent cell tissues like epithelia. So could you think of considering like skin cancer or something like that? Would that be appropriate to use the same kind of idea to try to...
With the vertex model?
With the vertex model.
I'm
just wondering, yeah.
Yes.
You would have like tons of data, I guess, also with shape indices and things like that, but I don't know.
I'd worry about the vertex model for that sort of data, because I think it's this two-phase stuff in a background matrix that we're really looking at here. So maybe looking at a vertex model where you have an active phase and a background passive phase, and starting thinking like that might be a direction to go.
Okay.
Yeah.
Just wondering.
But yeah, you're right. I mean, one of the next steps would be to try and do a similar setup with a much more cell-level model. We can't see what's happening inside the cell. We're not doing chemistry here. We're not doing genetics, right? So.
Thanks.
Yeah. But it's all very new, this. This is super unpublished, and I think we're going to fight with all sorts of referees with this one. Yeah.
Hi, just complementing the discussion from Francesco, if it is useful for biologists. I think a big question for biologists that work with cancer is the metastasis from the cancer, right?
Yeah.
So maybe it could be useful if we could measure when the cells are going for the transition to the from the epithelial from mesenchymal transition, and see if there is an activity parameter as our early warning signal, you know?
Yeah,
could be useful.
Absolutely. I think that's an interesting question. It's not obvious to me whether this big sort of initial tumor, does it explode? Or there's certainly work on small organoids where the cancer cells seem to creep out one at once, and I think that's more what's seen. We haven't thought of that in this, well, I mean, we have actually thought about it in other contexts, but not in this context, and there's loads to do, yeah. But we've got to be careful, because you can model this all you want, right? But it'd be really nice if it was useful for something, yeah.
Thank you, Julia. I have a simple question. When we decrease the activity in the last model, I think this have an importance on dynamical time scale, I think, because I wonder if we decrease the activity a lot, we start to see meta-stability behavior on carcinoid process, maybe?
What we did is we started with a huge thing and asked, will it break up? And certainly if you decrease the activity or increase the viscosity of the extracellular matrix, this single huge tumor does not break up.
I see, yeah.
As maybe you would expect.
Okay, so let's thank Julia again.